you're, 40 years out. Um, I will highlight one of our interns is one of your own now, Kira Battaglia. So I think she's the reason I was invited to even give this talk. But so thank you to all our interns, and also you're welcome, right? Like we've trained a wonderful generation that is now going out and making a difference in the world. So I feel like I've played a part in all of their success. And really, it's an uphill battle, and I share this, I think, in every presentation I give. But it's this quote from Aldo Leopold, who's kind of the father of conservation in the United States. And he says there are two spiritual dangers in not owning a farm. One is the danger of supposing that breakfast comes from the grocery, and other that heat comes from the furnace. And I know you guys are heavily invested in nutrition and policy, and so much of that is like is great, but when you realize that there's only about 2% of the American population that's directly involved in growing and producing our food, and that's producers, that's ranchers, that's all of it. And so it is it's wild to even think about. And so we do have an extra duty if you're aware of that need to really recruit and make it make it exciting, make the work that we do appeal to that next generation. Because this is, I think, the depiction that we get, right? Like you're Sisyphus, you are, and it's kind of exciting in some ways, right? Like because our work will never end. And what's unique about agricultural science is it can't just ever reach like a plateau and we can slow down. There's always a new challenge. There's always new opportunities to improve that, as the population continues to grow, we have new pests, new disease, human preferences change for the food that we want to consume. So it's like there's no stopping this boulder. And so we've got to push it for as long as we can and pass it off to the next generation. And that's yeah, maybe a little pessimistic. So there's like this other view that I really espouse because I work with fruit. And so this was our talented intern, Sam, who photoshopped me as an Oompa Loompa and Victoria Meekum, our molecular biologist, as Willy Wonka. Because really there's this like kind of fantastic opportunity to be imaginative and like really I don't know, really revolutionize how the food that we eat and the experience that we have as consumers, as producers as well. So I really hope that you can tap into that Willy Wonka passion for yourself, because really what we do is fantastic and so important to really the perpetuating the human race. Um, Unfortunately, when we talk genetics and plant diversity and germplasm, which is our lovely jargon, no one really knows what you're talking about. And I've gone and talked to plant breeding students, agricultural students, and no one really they don't really cover this. It's really loosely covered in undergraduate and graduate courses, but really germplasm is they are bearers of heredity, right? So exciting traits they don't exist in the next generation without germplasm. And so really any living resource that can contribute to plant improvement. And here's some lovely, beautiful examples of corn, carrots, tomatoes, squash. Really just that the diversity that, lights up our imagination, but also is critical to agricultural sustainability. And this so
Hi,
it can
Ben.
be, yes.
Sorry to interrupt. Can we just double check that you're sharing the slides?
Oh,
That you've clicked share again. During the setup, I know we turned it on, then turned it off to check. Did you
oh,
check this
Chris,
time?
you just let me go on forever and no one said a thing.
Sorry. My apologies.
Ah, that is painful. So
So apologies.
yes, as part of our duty to the generations, right, is to like jump in and say something. So here's our interns, here's all the slides that you missed, right? Like, so man, none of it made sense what I was even saying, did it? That's unfortunate. No, no worries. So we're caught up now. This is plant genetic diversity, right? It's eye-catching, it's spectacular. It really does like light up your imagination when you think of the possibilities and the food that we have. And what I love is crop wild relatives, and this is such an exciting field. Typically, we think of like interesting, I don't know, trait adaptations for climate or for disease resistance to new pests, but no, like flavor and like exciting production traits also come. Like the Concord, the grape flavor comes from a wild North American grape. So it's really, yeah, it's a lot there, and we'll cover some of that in this talk. so variation and germplasm. So it's this kind of this pyramid that we work at in agricultural systems, right? You have major cultivars and productions. They're sitting at the kind of the pinnacle here, the peak of the pyramid, and it's really narrow. There's not a lot of diversity there, and that's kind of by design because again, we've told we want to diversify our functions in society. We don't all want to be growing food and producing food for ourselves and our communities. So we've outsourced that. We have specialized community members who do most of the production, and they can't grow it all. And that's dangerous because new pests come in, systems change, you outgrow a system, but the crop genetics remain the same, and now you don't have that flexibility. So we need to go down several layers. So I work kind of at this bottom, the base, where we have lots of genetic variation, a lot of interesting potential, but a lot of bad quality. So things you would never want to eat, but they might have some potential there. And so they move up in a breeding program to this middle layer where there's like some elite things. Most of the world never gets to try those because they're just not quite good enough to get into producers' hands and then go on to that next generation of consumers. But this is the bottleneck. And if you studied population genetics, right, like human domestication of crops and evolutionary genetics works in very similar. And a lot of what we understand about, how evolution works in nature comes from studying human domestication events. So this is, you have a bottleneck where you've lost genetic diversity, and there's really no way to go back to reintroduce it unless you have that source of germplasm. So this is a view of the apple collection, and so we have, close to 2,000 cultivated apples. Some of them are wild, but cultivars named varieties of apples, close to 2,000. But if you look or understanding this like kind of this plot here, each line represents a first degree relationship. So it could be a parent or an offspring or a sibling of yeah. So kind of understanding their relationship among each other. And you see this really tangled web. So when we study like apple and how much, like what is the real breeding population of apple in modern cultivars, it's only about 8 individuals that have contributed. So of the thousands of possibilities, we've really narrowed down the genetic variation. And you can see that in the store where a lot of the apples, they look and feel and taste similar. And that's great because it helps uniform production and standardize qualities, but then you're missing the element of excitement, right, for consumers as like new crops come out, and you're also missing that flexibility if a new disease or pressure comes out or we lose land, right? Like more people, less land for growing crops. Now we need to minimize our footprint. So I'm part of a larger network of gene banks across the nation, and this is called the National Plant Germplasm System. And so our mission really is to acquire, preserve, characterize, and distribute plant genetic resources. And as of today, we have over 500,000 individuals conserved in our gene banks, and that's really exciting. So we're here in Geneva, New York, and then Davis, California, Corvallis, Oregon, they are kind of our partners studying fruit because most of our gene banks are for seed preservation. That's how most of our crops are managed and handled. And this is really the view that you get. This is the Svalbard doomsday vault, right? But most gene banks just look like kind of a boring annex in a library. So stacks, that's refrigerated. You should add that, I guess. So a lot of these are just tucked away as seed. Most really can't be explored or evaluated because you have 500,000 accessions tucked away as seed. You can't realistically pull them out and grow and explore the collection. But with our fruit collections, which can't be stored as seed, which need to be preserved as trees or individuals in a field, as vines and whatnot, so we have this like living, 100% accessible, 100% vulnerable collection growing here in Geneva, New York. And we've got, companion sites in California and Oregon. But we do have one of the world's largest apple diversity collections here. And it's such a national treasure for us because it represents worldwide diversity of apple, and apple's so beloved for us. Grape is actually split between two sites, California And Geneva, we have the cold hardy material. They have the European grape and our beloved tart cherry, which is another cold hardy relative of sweet cherry. And when I say vulnerability, I really do mean vulnerability. Like the we have disease, we have pest pressure all the time, kind of infiltrating the collection. So this, if you can see my cursor, or maybe not, but the little plot on the left is showing the kind of an overview of the collection. So each square represents a tree, an individual, and it's genetically distinct. And the color represents severe severity. So in 2020, during the middle of a global pandemic, we had an outbreak of fire blight. And Kira was here for this too. And I think she left, I think maybe that's why it happened, because she left our unit at that time to go pursue other things. So yeah, that's interesting connection there. So in 2020, yeah, so we had this outbreak of streptomycin resistant fire blight. So our typical chemical controls had no effect, and it just swept through the whole collection. So disease severity of 6 is pretty severe. Like you're going to have to probably remove the tree at that point. And you can see how much purple is there, indicating really high severity. But excitingly, you see a lot of yellow because either these are lucky escapes, or as in the case of the picture of the two trees, you have one really healthy, robust individual next to a pretty infested individual. And that's just somewhat of the natural genetics of disease resistance. So it was a wonderful opportunity to evaluate the collection for disease resistance. And you could see it really ranged from almost 100% infection to 0%. And some of that really does align with what we already understood about the species and the genetics of fire blight resistance. So I mean, it's just a change in management, but still, these collections are vulnerable to disease, to a tractor getting loose, an intern makes the wrong cut, and they can destroy an accession that's irreplaceable for the future. Unfortunately, some of our chemical controls of control fire blight really impact fruit quality. So now these are things that are still edible, but if you're a grower, a producer, these you can't sell these. And so maybe a farmer's market, you might make a little profit, but for consumers who want flawless fruit that's grown sustainably with minimal pesticides, this is what you're up against. So you need really strong genetics to control fire blight, which not only impact the health of the tree, future production, but also the consumer aspect. So because of that vulnerability, we have a method called cryopreservation that we actually now partner with Fort Collins. We used to do it on site, but this is taking dormant wood, which is used to propagate or to graft, and you can essentially dehydrate it and put it into liquid nitrogen, which is very cold. If anyone's worked in the lab with it, yeah, you know instantly the danger and the severity of it. But this tissue is able to be plunged in liquid nitrogen and stored indefinitely. And so we do this work routinely. So we have about 90% of the collection backed up in cryo, which is wonderful. Some of our crops, like grape, is really challenging because you can't do direct cryo of dehydrated wood. You have to do shoot tip rescue. So you have to not only initiate it into an in vitro, like a tissue culture collection, and do droplets of cells preserved in liquid nitrogen, and then followed by rescue and tissue culture, which is it's a whole mess. Because you might get contamination, you still might have really low recovery rates. So the grape collection is very vulnerable to losses in the field, and grape is one of the world's most valuable fruit crops, mostly for what? For wine. I keep mentioning our partners in California. They have a gift of warm, beautiful, sunny weather, whereas we're like 9 degrees in Geneva today. It's pretty miserable. But that gift is that we have is dormancy. So that cold weather really drives dormancy and allows for cryopreservation. So we brought all the plums that are normally maintained in California to Geneva um, expressly for that. So we can get them cold exposure, grow up material, and preserve it in cryopreservation. So 144 plums made their way this year to Geneva. And so now we can evaluate their cold hardiness. So yeah, not only back them up and make sure they're safe for future generations, but now evaluate, cold hardy adaptations. Most of them probably won't do great fruit production, but there might be a few that stand out, and we can get those to our growers in New York. And this is so fun. If you've never done this, this is one of the world's oldest hobbies. 10,000-year-old technology of just taking your favorite plant from a forest or wherever, grandma's backyard, and putting it and propagating it. And so we do chip budding, which takes a really small section of a viable bud tissue, which is going to sprout out next season into a new branch, and you can attach that to a rootstock, and you can successfully graft and clone a tree 10 millions of times. And that's what's been done in all apple production, and especially modern apple production. So we can, you know, really, like a tree, an apple tree might live 100, maybe 150, 200 years. A cherry under maybe 80 is the kind of life expectancy. But through grafting, we can perpetuate and keep an accession, an individual alive for generations. And that's really cool because we have this timeless aspect to what we do. So Roxbury Russet is like the first American apple. So we typically think of apples as American, but early settlers brought them over. A lot of French influence in the apples that were initially brought over, which is kind of interesting and surprising because we always think of like, oh, the English, they brought over their apples. But it was a huge French composition that we're finding. But Roxbury Russet was the first one that was really unique to America. This wasn't really bred, or it just naturally kind of just occurred through a chance seedling. And we've harvested that, and we're perpetuated that since the mid 17th century, which is cool. Montmorency, which we'll talk about a little bit, is a tart cherry from the Montmorency region in France. Again, it's about 400-year-old cultivar. Concord, which everyone loves. This is like the kind of classic grape flavor. This is a late 19th century just random chance selection that a farmer made. And so really, like, you think about how these, I don't know, this timeless aspect of what we do, because you can really see how apples have changed, how our crops have changed over the centuries. And what was really exciting, kind of this is the call everyone hopes for, except for me, it wasn't like you think I'm going to be an archaeologist one day. I'll have a chance to like dig in the earth. We got the call from Mount Vernon because they had discovered cherry bottles preserved pre-Revolutionary War in Mount Vernon. So Mount Vernon is George Washington's private estate. So this is where he spent a lot of time before the war. Um, and they found two bottles initially that they kept digging, and they found 35 containers. Some had gooseberries or ribes, and but most of them had cherry remains, which I mean, you can see here there's like pulp, but mostly it's pits that are still intact. And this was fantastic. And so learning with the archaeologists, this was their dream too. It's kind of funny because the archaeologist said he always knew he was going to find something fruity in his life. Because people have found pits in other digs, but never the extent and volume that they found here. So what they identified was a pre canning. This is before modern ball jar, mason jar canning with like heated material preserved, right, with wax or whatever. These were um, cold stored, cold packed cherries that they put in old wine bottles, and they dug clay pits in underneath in the basement of Mount Vernon and preserved. And they were meant to last about 1 year only. And so these were then buried in a layer of brick and then later cement, so they can really date well. And that brick and cement added to the level of compaction and preservation. But we have a chance now to work with Mount Vernon to understand what this cherry might be. So that's a picture of me and Tori in the basement of Mount Vernon. That was an exciting opportunity. But we're finding stem fragments. We found like intact fruit. We also found like cuticle peels, which is really cool. So this is like just a microscope image of stomata, which are like the breathing structures. So a lot of stuff is still intact, and you can explore kind of the agricultural or horticultural qualities. And the kind of ultimate goal is to identify using genomics to kind of match what might be in existence today. And as I keep mentioning, grafting, right? So a lot of what George Washington had planted and had documented in his own notes are cultivars that still exist today. And so there is a chance that we might still have this material today. And here's kind of a zoom in on one of the pits that we kind of cracked open. Pits make great resources for DNA. And then here's the seed inside. The seeds are not likely viable. They're pretty waterlogged. So it's a chance of regrowing from a seed is minimal, but if that cultivar has been preserved through grafting today, that would be very exciting. And again, here's the Washington quarter for comparison, size comparison there. We believe it's a tart cherry just based on our initial genetics. It's kind of matching towards more of this like tetraploid. So tart cherries is complex because it's got two copies of each parental chromosome. So it's actually a hybrid between sweet cherries and a European dwarf cherry. So really different fruit quality. Tart, as the name, like highly sour, much more acidic than our sweet cherries. Much more gummy in texture, and they have an interesting nutritional quality to them because of the anthocyanin composition is a little bit different from a sweet cherry. You're getting a little bit more of a nutritional boost. Evidence of higher melatonin. We have a nice method. So hopefully in a few years we can have that data published on melatonin content, and then like made famous really by TikTok Sleepy Girl mocktails. So thank you, Sleepy Girl from TikTok. So try it out if you're having trouble sleeping. But tart cherry juice is kind of it's having a moment, or maybe it's had its moment and maybe it's moving on now. But really not that much different from prune juice. But grandmas drink prunes, right? Like grandmas are drinking the prune juice. Elite athletes are drinking tart cherry juice, and it's really for that sports recovery promise with the high anthocyanin, high phenolic, high antioxidant content. So this is just an example of some great branding. Works great for me because I do love tart cherries and want to see them more out into the community. The downside with tart cherry production in U.S. is it's 90% dominant with Montmorency, that 400-year-old cultivar. And as the world changes around, that because of clonal preservation, that there's no way for Montmorency to change. So new traits, new adaptations, it's kind of fixed in time, which is fantastic if you're doing like studies and comparisons, but bad if you're really doing production and want to change methodology. If consumers' preference changes, Montmorency has pretty low anthocyanin content, and so if athletes continue to want higher content of nutrients in their tart cherry juice, really you have to look beyond Montmorency. So what I'm going to talk about briefly here is our gene bank characterization. So we look at phenology, which is kind of nature's calendar, like the seasonal recurring events, bud break, bloom, fruit harvest, dormancy, and then some fruit chemistry traits that I love this area so much. So tart cherry bloom phenology. So when we talk Montmorency, Montmorency typically is kind of I don't know, how do you say that politically? But anyways, it's an okay crop. But it has a tendency to bloom a little bit, break dormancy and bloom before you're free of that Midwest or the spring frost, which is common in New York, the Midwest, where tart cherries are more prominent. And so you'll lose an entire crop because you've got this monoculture, and you've done nothing to diversify how much is grown. So we've looked at bloom phenology, which if you can bloom later, that's a little bit better for colder climates. It's not advantageous for warmer climates. You still want genetic diversity for um, earlier bloom because then you can get your crop out there into the market a little bit earlier. But for New York, for Michigan, where a lot of these are grown, you really want a later bloom. And so evaluating the whole diversity collection, and you can see there's some variation, Pleuronus thoracis, you can see some variation for how much growing degree days or how much heat is required to make them bloom. And so you want more growing degree days. You want higher loads. So some of the wild species that we could bring in as genetic resources could hopefully extend that as well. Grape in New York is the same problem. It's not about cold hardiness because they do great when it's dormant. They can handle anything you throw at them. But as soon as you get a little bit of warm weather, like we had a 40, 50-degree day in January. Some days we have like a 70-degree spike in late January, February, and that's going to start vegetative growth in our crops, and that can lead to some frost damage because you've lost dormancy. So delaying dormancy as long as possible. And it has a really interesting environmental and genetic component. And so here's just a kind of a quick plot looking at Vitis vinifera, which is the European grape, and you can crossbreed it back to wild grapes. Um, Interestingly, like the more vinifera you have, the longer it takes you to bloom, which is actually now we were talking bloom and not bud break. You want to increase the or decrease how much time it takes to bloom in cold environment. Kind of inverts because grapes take so much longer after the last frost to set bloom and start fruit production. So again, it's this genetic component at play and something growers and breeders are need to be mindful of so that they're making accurate selections for their growing regions too. What's really cool is that you can use temperature or these kind of physiological techniques like the low temperature exotherm. Differential thermal analysis allows you to freeze living bud tissue and find out and measure the exact point at which it dies, which is, really useful for us because we want to know what that max is. So you can take bud sections, throw them in a programmable freezer, slowly lower the temperature until you can measure cell death. And so these plots are showing the rate of how much cold a cherry can handle over the course of the winter. And you want to see it really dip. That's what makes physiological sense. You're seeing it enter a period of dormancy and then leaving dormancy as the winter ends. But each year is so erratic. So I'm showing you 2 years, 2 seasons of data, and 2022 to 23 was so erratic. I mean, they started cold hardy and then they were just bouncing all over the place. And so the genetics and environmental component can get really complicated as you try to do these assays. But imagine you're a grower and you're trying to set up a 4,000 acres of production, right? Or something outrageous. And you've got to decide which cultivars to use. So we need this data to accurately help our growers or breeders, make these selections that will best accommodate their growing conditions. On to my favorite though, fruit quality. This is what we all care about. Like, because this is the part if you forget that this is where we connect most with the fruit, not the bloom, not the vegetative stuff, which is interesting and important for growers, but consumers are drawn to the fruit quality. And here's just kind of a nice example of fruit genetic diversity in apple and cherry. And I mentioned cherry anthocyanins, which are the color pigments, but also the antioxidants. The kind of horizontal or the vertical line represents Montmorency, the industry standard, and it's on the extreme low end for anthocyanins. Some of ours are kind of through the roof with high rich anthocyanins, but nothing quite like grape phenolics. So when we look at grape phenolics, cherry has maybe two prominent anthocyanins. Some of our grapes have up to 30 unique compounds that we're measuring. And so you're really diversifying the anthocyanin profile. Apple's even more sad, and I won't even share that that data because it's just one major anthocyanin that contributes to color in apple. So pretty poor for phenolic composition. Aroma, though, this is where you really can feel like Willy Wonka, right? Like that everlasting gobstopper. Some of our grapes, benzaldehyde is kind of the prominent grape or the prominent cherry almond flavor that's in our that cherry and almonds, right? And like they're interchangeable, and depending on the setting, you can kind of trick your brain into tasting one or the other. Um, but we measure benzaldehyde. Some of our cherries have almost no benzaldehyde, but they've got this linalool, which is much more floral and aromatic, similar to a muscat grape, which we'll talk about in a second. But benzyl alcohol, benzaldehyde. So this is our techniques are able to kind of measure what we're tasting a little bit. It's not great though, because all our olfactory senses are so complex and dynamic. We can give numeric values to a lot of these compounds, but in terms of human preference and what we can detect, what we actually enjoy is a much more complicated story that once we have this data, we can work with maybe some taste panels and get some more human data on that. But Foxy aromas, this is the Concord grape. The purple compounds are the ones associated with Foxy grapes like Concord, phenylethyl alcohol. We see that also in grape, in cherry as well. Muscat grape, though, this is such a unique flavor. And actually, I think we took Kira here. to Davis, California, and she got to taste a lot of these interesting stuff. So some of this data came from material that she helped to collect as well. But I think that's the future for most of our crops is interesting aromas that's really going to draw in the consumer. So linalool is one of those that I would hope to target more. Um, skipping over that estragol licorice flavor. Who doesn't want a licorice flavored apple? It's a really kind of uh, surprising to taste, and it's something that develops certainly with time. So like standard cultivars like Golden Delicious really enrich in estragol the longer you wait to harvest. So pausing a harvest to get the that licorice crowd excited about eating some apples, that would be fantastic. So here's Honeycrisp, has really kind of a much more generic kind of classic apple profile, really low estragol, so you're not getting a whole lot of it of that. But this is alpha farnesene, which is commonly associated with the storage disorder. So you'll get apple scald just by having this high volatile content. So some of what we do look at aromas not just for human consumption, but also
Mhm.
for the storability. And what do we do? What do we do with all this data besides publish? We have a database where you can research and evaluate for yourself and all of our traits for not just apple and tart cherry, but for any of the crops in the National Plant Germplasm System. Just as I close, like I know you're not maybe working with plants directly, but like you've got your own form of germplasm, right? Like you've got whatever you do in your career, you've got to maintain some level of diversity in thought or in resources so that you can stay flexible. So you don't, no one wants to be painted into a corner. So that's just my plea is like consider your germplasm. Like consider whatever it is that like I think so many studies that I evaluate, they jump to conclusions because they didn't have the right set of starting material to begin with. And don't lose that magic of why you even got into this field in the first place. Highlight again our public tours since I wasn't even showing the slide before, but Saturdays. So I can share these dates with Chris and he can get them out. If we'd love to have you. No fee, no tickets, just show up. And just thanks to wonderful groups and thank you all for having me. Whew, that's a sprint to get done in a half an hour. And I know I'm a little over. But now I was promised good discussion from you all. So I did my part. Now you're on. And Chris said I could just call on whoever I want from the audience too.
Uh, yes, thank you once again for a terrific talk, Ben. Please feel free to raise your hand if you have a question in person, or I will be monitoring the chat online. So please feel free to type it in the chat. I see we have our first hand raised. Here you go.
Thank you. Really, really interesting, really fun. Um, I was wondering if you could talk more about kind of the challenges of breeding for climate change, because a lot of what we're seeing isn't just like, it's not predictable. Like that's kind of the nature of
Yeah.
it. You're seeing more big swings even within a season and year to year variation as well. And so what kinds of things do you look for when you're breeding specifically like trees, like long-lived crops for these kind of really erratic types of new seasons we're having?
Yeah. So with Apple, it's exciting because you have a rootstock component, which really can dramatically impact its climate adaptation. So in terms of disease, you can slow down growth to have um, much tighter wood growth, so you're much more resistant to disease pressure. You can delay bud break and bloom just by the rootstock that you choose. Uh, but here's the challenge, right? Is that historically each region had their own unique apples that they produced. I have a whole book on my shelf of southern apples, and they were only grown there because that's the only place that they would grow, and they were proud of that, right? But now we've globalized our markets. So now we want to grow the same cultivars in New York that we're going to grow all over the country, and that's just yeah, it's not feasible. It's great for planning and controlling a market, but not really ideal for kind of prioritizing and doing the best you can with what you've got, right? So I would love to see, smaller growers take on maybe more diverse crops, but it's up to consumers too, right? To try something new and not stick with old cultivars just because they might be your favorite, but if they're not, you know, I'll stop there before I say too much. I can get into trouble naming cultivars and seem like I'm being biased against some. Does that make sense though? Is that clear?
Yeah, I mean, I think that's helpful. I think what you're saying is that like having more different kinds of varieties within a region is helpful.
Yeah.
Because I was just kind of looking at, so you're like showing the bloom date and the cold hardiness phenology, and it's kind of clear what you are looking for a given region if you know what the future of the weather is going to be like in that region, but we just don't know. And so,
Yeah.
you know, so the answer is just grow lots of different things. Is that kind of your answer to that?
That's my simple answer. Yeah. So some of the research that combines the physiology with the genetics is looking into like how do you delay bud break with kind of a chemical application that you can put on. So if growers know a bad winter spell is coming, but their vines or trees are losing dormancy, something that they could chemically spray. But then the challenge is each individual responds differently to that application. So there's no silver bullet for breeding for ideal climate conditions. Which is again, that's Sisyphus, right? We get to keep pushing the boulder. As long as we have challenges, we can meet them if we've got diverse genetic resources.
Thanks so much for a great talk. You talked a little bit about sort of the popularization of tart cherry in the context of their health profile and kind of catching a fad or a trend. I'm not
Yeah.
sure if you have a read on this or a thought based on people reaching out to you or things that you've been seeing. Do you feel like there's more interest in growing these unique varietals that maybe have a better health profile in smaller, more regional farms? Or is it is that like not necessarily enough of a driver right now, or the information that y'all have and that you have on the database is just not getting out there as sort of a health spin on these different, maybe more diverse crop varietals?
Yeah. And that's a really complicated question and a complicated answer to so I didn't even mention a huge component of what we do is distribute these genetic resources. So we do actually work with a lot of small growers, small producers aimed at like farmers markets. And so they can get these unique varieties and cultivate them for their own unique niche that they've found. But in terms of like how the global markets, right, even within the US is responding to the need for broader genetic diversity, broader nutritional quality. I know there's been a new push with the new food pyramid, right? Like with like let's focus on whole foods and things like that. But I think it's still pretty nuanced and it's hard to make a dent in kind of the big chain markets, right? Like there's only so many brands of tart cherry juice that you'll find on the shelves, and other health products derived from plants. New York is unique in that we can find maybe, geez, 10 varieties of apple at Wegmans, which I know some states, they're still down to the color system, right? Like, do you want a yellow or green or red apple? And so I do see some areas kind of specializing and highlighting the diversity and then the need for unique resources, but yeah, I don't think it's uniform or there's not a big push from companies at least.
That's great. Thanks so much. And you said that you provide some of those varietals. Do y'all have a way of promoting small growers to grow these varietals, or like grants that y'all have through the USDA or otherwise?
There might be through our larger, because again, USDA is huge. We're just this very small program within it. So we do have ways of like connecting to growers and they find us certainly. But in terms of like opportunities to help them grow or grants to grow, I don't know. That's a great question. I'll study up on that.
Thank you very much, Ben. Fantastic presentation. I'm Yelena Naumova. I'm teaching classes on data visualization, analytics, AI, and whatever data you show, it's like a golden mine for data
Yeah.
visualization. It's salivating, it's color, it's taste, it's aroma, it's everything all together. But I know that this data are really very difficult sometimes to find. The proprietary owners are not eager to share with this data at all. And I believe this is a fantastic teaching material and something which would be really very useful for distribution, for better communication of what you are doing. So I'm wondering if there are sources which are in the public domain, easy to extract, easy to download, and to work with this type of a data much wider than what you have and help you out with commercialization of your products.
Yeah, that's something that we as an organization discuss a lot. And so typically, I mean, we are sitting on gold mines. And so our goal is to, certainly as a scientist, I hope to publish because that's the easiest way for people to not only find your results that you even worked on a project, right? But then to kind of validate the methodology that you did. But a lot of us also collect data that just immediately gets put into GRIN, which is that main database. So I'd love to, yeah, talk more. So if I could get your email number, I think Chris has mine, he can share it. But yeah, we would love to talk more about integrating AI resources to do kind of really large, complex data mining for a number of crops.
I enjoyed the comments about cultivars and the history, just because they're have such a long history for a particular cultivar. And a couple of the names you came up were Eastern Massachusetts names, and I wondered if these were literally the location. So Concord Grape, I wondered if that was Concord, Massachusetts, and Roxbury.
Okay.
And it might seem an odd question, but I wonder if you have any favorite sort of agronomy history tourism in Massachusetts out of this area of interest.
Oh, that would be, yeah, that would be exciting. So there's a main heritage orchard, and they're kind of the leaders for apple history and cultivation from the Northeast. And they've done extensive work. Todd Little Seibold does so much. He's the one that really discovered the French influence of apples. Because again, we're thinking the English brought the English apples, right? Like no, it was primarily the French and their contributions made the biggest impact on the domestication of apple in the United States. But the agro-tourism, people come, yeah, from all over just to taste, a Russian apple from their childhood. Or, it's a big field that we could explore. And I do like to think that we have kind of the ideal classroom for that. We have not only if you're studying genetics or physiology, agronomy, but if you want to study the history of cultivation, right? And I think it was Lexington, which is where Concord's was developed by Ephraim Wales Bull. I don't know why that guy's name sticks out, but he was the one that discovered the Concord grape. But you're right, like the names kind of denote kind of their place, and I think that's why the Mount Vernon cherry is so interesting and compelling a story because I mean it's a cherry. Like we've got plenty of cherries, but something that connects to like such a I don't know, prolific American leader, and anything that ties us back to our foundation as a culture, as a country, like is tremendously exciting. And not just being American centric, but all cultures feel this way and have visited the collection with that in mind as well. But no, let's partner. Let's do some tours of Boston and area and do an agricultural, yeah, a tour of the area, not just the rich culture of Boston and Massachusetts in general, but just tie in the agriculture part.
That sounds super fun. We have one more question in person, and then I see two online from the chat and one member with their hand raised. So we'll start with the in-person question here.
Hi, Ben. Thank you so much for that really fun presentation. I have a question. I'm curious about what happens once you've bred a varietal and then you've bred it for whatever purpose and it's great. What happens? How does it get from there to other farmers in the country? What is the ownership process for that? And yeah, how does it become available to people?
Yeah, that's gotten so complex, especially with intellectual property. And anything out of a university, the universities want to, strictly control those things. So like the example that I have is my grad advisor at Cornell Susan Brown released Snapdragon, which I don't know if you all have had that one yet, but it's a Honeycrisp offspring with really enhanced quality, like even better crispness, better flavor, better storage. A lot of the problems of Honeycrisp are done, are kind of resolved with the Snapdragon. And there'll have to be a new Snapdragon eventually, but Snapdragon was released as a club variety. So growers had to buy in to get access to it. Because again, you want to control quality. If you're using a uniform name like Snapdragon, you want to make sure that all growers have equal contribution and stake so that you're not, delivering garbage apples that are going to hurt the brand. And so that was one avenue that Cornell took. Susan Brown released 4 additional ones, just public releases, so anyone can buy them and have access to them. Washington State has their Cosmic Crisp, which was done similar to Snapdragon. Again, you had to have buy-in, and it's strict controlling of the market because you can overgrow a crop and then really decrease its value. So some coordination needs to go into some of that. But I think some of the public releases are ideal if you're just looking for something great to grow in your own backyard or a small production.
Thank you. If you don't mind, Ben, do you think you could please read aloud the question in the chat and answer?
I'd be interested to hear your thoughts on advocates such as Dan Barber who focus on breeding for flavor. How do flavor traits compete with practical traits like shelf life and the availability to travel well? I'm also curious about your views on trademarking specific varieties. Thanks. Yes. So I think we saw with like the tomato industry is we bred the joy out of tomatoes. If you've ever had a garden tomato, it's life-changing, right? Like especially if it's at grandma's house and it's magic, right? And then you go to the store to recreate that and you get nothing. It's just you paid for red, I don't know, pulpy water. Sorry, I have got strong opinions about that. But so that was just really to increase the growability of the crop, not necessarily thinking about the consumer. So much we've grown as in the research community to better understand the genetics of what the growers want and now the genetics of what consumers want. And so with genetics, you can really make quick selections for disease, certain production traits, right? And not just thinking about apple, but really like apple, the fruit is kind of the latecomers to the game, but most of our major commodities, like genetic resources or genetic tools, genomic tools to make those quick selections so that you can be freed up to focus more on quality, which is, I think, where we need to, that's where we've let the public down, I think. So yes, yeah. Trademarking. So apples are sold by cultivar, which I think makes them, yeah, we love, it's like Pokemon cards, right? Like each one's unique. We love to like know the apples. Other crops, they don't necessarily trademark, right? They don't have like, you just buy Driscoll berries. You don't buy necessarily the cultivar that's in them, which there is a cultivar in them and it's distinctive, but um, necessarily not trademarked in that way. I think it's important, but also limiting because people get fixated on a cultivar, and you see this in the wine industry, right? Like we want to be growing Merlot and certain cultivars. Maybe we've outgrown them in terms of our production, and maybe they're not sustainable, but we know the name, so we want them still. So I think trademarking can hurt an industry and also save it in some ways. I mean, it's really great that like George Washington like knew the varieties that he was planting so we can kind of trace that back. That makes it great for like accountability and knowing what people were doing. But otherwise, yeah, it's a double-edged sword. I see Nick has a hand up.
Yeah, thanks for the presentation. As everyone said, I was just curious about what, if any, like international collaboration y'all do in terms of finding new varieties or trying to find things that are climate resistant or whatever. And in particular, I was curious with, for instance, I know like apples, I believe they're from Kazakhstan, like their
Yeah.
origin point. If there's like any kind of collaboration y'all do with trying to find those like hot spots for genetic diversity, or if y'all are more trying to focus just on like historically relevant ones to America.
Yeah, that's a fantastic and a really key part of what we do is like, so we talk a lot about cultivars because they're named and we have a connection to them, but the wild relatives are the most at risk. Urbanization, right? Like just my one of my favorite apple species is used as firewood in some parts of the world, and so it's like that's no, don't hack it away for firewood. Get the germplasm, get the material, and then use it for firewood. Like we still need to use the resource, but let's make sure it's conserved. So that's become increasingly complex over the year. We do have international treaties to make sure like both parties, both the recipient and the donor are being protected, right? Because these are their unique resources. Kind of gone are the days. Like in the 90s, we helicoptered into Kazakhstan. We as in our unit, now I wasn't even around by then, but they went in helicopter to the mountains of Kazakhstan and collected wild Malus sieversii, which was critical because the deforestation has gone on the last 30 years have really reduced those populations. So it's key that we get in and we're partnering. But I think more than us holding it, it's that international partnership. And that's what we're trying to move towards. It's like you do your job conserving your stuff. We have native North American species. We can be the gatekeepers for those, and but making sure that we're partnering and sharing resources and information as needed. That's the ideal. But yeah, because there's so much, and most of it's out of the United States for apple and for cherry. There's a lot of native prunus species, but most of the exciting ones are in Europe and Asia.
That
Um,
is great. We have one question in person, and I see Isabel's hand is also raised. So if there's time for both, that might close us out for the day.
Thank you. Thank you, Ben. I'm glad you're able to come. I would like to know, what is your favorite apple cultivar and why? And what is your favorite tart cherry cultivar and why?
oh man, that's an impossible question. It's like, yeah, it's like asking your professor, who's your favorite student, right? That doesn't work. Except in private, they'll tell you. So no, I would the one that really excites me is this one called Cur, not Cur, like Kerr, and it's a Siberian hybrid apple that has like a unique tropical flavor, and it's small, like it's maybe just over the size between a golf ball and a tennis ball. So for those like you're on a long hike, you don't want like a giant pumpkin apple in your bag. You could have a kind of a handful of these, and they really do have an interesting flavor. Tart cherry, that's even harder because there's so much flavor and exciting things beyond just Montmorency sour sweet. So I'll get back to you on that one. But try Cur or come to Geneva and ask for Cur, and we'll see if we can deliver.
Okay, I will. Thank you.
Isabel?
Yeah, thank you for your passion and excitement about flavor and food. For all of us people that are super passionate about food and flavor as well. So I recognize that these are cultivars and not necessarily like mainstream industry level varieties. But I'm just curious to hear your thoughts on changes in nutrient levels in either the cultivars that you work with or in general over time. Since you kind of talked about flavor declining in tomatoes. We know that flavor compounds are actually linked with nutrition as well, so I'm just kind of curious to hear your thoughts.
Yeah, yeah. So a lot of the studies in apples for phenolics have shown that they've decreased somewhat over time. So the complexity of phenolics has really narrowed down. And I don't know if that was intentional or not, because I mean, phenolics can add some bitterness, especially if you get those like those tannic compounds. But for the majority of the small molecular weight phenolics, really just in enliven the color and I don't know. Yeah. So I'm not sure why they were selected out, other than just we were using too narrow of resources. Breeders are more mindful of that now, and so as soon as the beauty of bringing in trait qualities like disease resistance, adaptability from wild is you immediately get an infusion of interesting phenolics. Like the apple I was mentioning, Cur, is a hybrid between two apples, a cultivated and a Siberian crab. And Cur actually has a really kind of unusual, kind of a purplish hue to it. It's distinctive from like this, the classic red. And some of it's like the waxy bloom that the apple gets on the surface, but you do get a much richer pigmentation and complex phenolic profile just by that hybridization. So I would certainly love to see that. I think the challenge is if phenolics, if we understood phenolics as well as we do vitamin A and like our major nutrient groups, right? Like, because there's still, we know that there's a health benefit, but it's not like something magical that we can say like you're gonna, stop blindness, right? Like with carotenoids. So I don't think there's a tremendous interest to select for kind of phenolics alone. I would love to just explore that area a lot more because I do think there's so many that these crops produce that we have no idea what they do. I skipped through caftaric acid, but that's really high in some of our grapes. And then doing like drought studies, you find that caftaric acid in vegetative tissue, which is correlated to fruit quality, like has really it really caftaric acid is correlated with increased drought tolerance. And we're seeing it high in the fruit, high in vegetative tissue. Like, what is it doing there? And what is there a benefit also to humans as well? So yeah, it's an area that I think needs a lot more work, a lot more students. Maybe we could get a few of the whiz, students from Tufts on this problem right there.
All right, please everyone join me in once again thanking Ben Gutierrez.
Thank you for having me. Again, sorry, not in person. Come to Geneva.
I have a quick announcement to make. Our next speaker series is on February 4th. In the intervening week on January 28th, Will Masters will be headed to Stockholm to receive a prize in global food security, and his talk will be at the speaker series the week after, February 4th. We'll be giving a full lunch to all those who attend instead of the usual refreshments. And we'll ask for that reason you please RSVP. Information to RSVP will be sent to your emails shortly. So please keep an eye out for that. And we're excited to see you all again for our next speaker series on February 4th. Thank you.